The MethylFlash Global DNA Methylation ELISA Easy Kit is a simple and convenient way to rapidly screen global levels of the 5MC modification. The assay with this kit can be completed in as little as 2 hours, and results are comparable to HPLC analysis, reflecting true status of sample DNA methylation. In the assay, DNA binds to specifically treated strip wells. The methylated DNA fraction is detected using capture and detection antibodies, and then quantified colorimetrically by measuring the absorbance in a microplate reader. The kit contains all essential components needed to complete the assay. Begin by preparing the DNA standards. Use microfuge tubes to dilute the positive control DNA to generate different concentrations, as specified in the user guide. Arrange the strip wells in the plate frame. Add binding solution to all wells. Then, pipette the prepared standards into duplicate wells, along with the negative control to establish a standard curve. Next, pipette your DNA samples into duplicate wells, arranged in a similar manner as the standards. Gently shake the plate to mix. Cover the plate with the provided adhesive film. Place into an incubator for the required time and temperature to allow efficient DNA binding. During the last few minutes of incubation, prepare the 5MC detection complex solution by diluting the 5MC capture antibody, followed by the detection antibody and enhancer, as indicated in the instructions. Remove the plate from the incubator. Wash the wells with diluted wash buffer to remove unbound DNA, repeating the wash step as directed in the protocol. Add the 5MC detection complex solution to each well. Cover the plate and incubate at room temperature. Once incubation is complete, wash the wells again to remove unbound detection complex. Add the developing solution to each well, and monitor as a blue color develops in the positive control wells. When the desired intensity is reached, add the stop solution to each well. The color will change from blue to yellow, and the plate is now ready for measurement. Read the absorbance values at the recommended wavelength. Use the values to generate a standard curve from the control DNA concentrations and calculate the percentage of 5MC in your DNA samples. With the MethylFlash Global DNA Methylation ELISA Easy Kit, you can achieve fast, sensitive, and reliable quantification of DNA methylation. Please visit epigenetic.com or contact your local distributor for more information.